as the posterior pituitary or a neurohypophysis, whereas the anterior pituitary comes from the stomodium of the entoderm. So there are two different formations within the pituitary fossa. Altogether, we call this shortly pituitary or hypophysis. And again, when we just look at the overall number of the pituitary adenomas, when we just put their location among the all intracranial mass lesions, they form 10 to 15% of all intracranial neoplasms. It totally depends on what kind of data registration you are looking at. For example, the previous two institutions that I worked in, both in Marmara and Acıbadem, the neurosurgeon in that particular department, that's the same guy, was very curious about the pituitary the surgery. So these are the total amount of the number of the pituitary cases for about more than 22 to 24% of all cases. So this depends upon at which kind of registration you are looking at. And this data comes from the 1998, the Sylvia Assa and the Shirin Azaz, the Azaz description. And they are accepted as benign lesions, but some in parentheses show an aggressive clinical behavior. That is to say that they have the habit of invading the adjacent tissues, and they show a high proliferative activity in terms of the mitotic. It means that they are mitotically active, and this can be easily seen under the microscopic evaluation on hematoxylin and eosin stains, shortly H&E stains, as well as by using the specific immunohistochemical techniques known as the Ki-67 or MIB1, the staining procedures. And on that slide, what I'm just trying to show you is the some specifications about the pituitary tumors. This is to say that we divide the pituitary adenomas as upon their dimensions. If a pituitary adenoma is less than 10 millimeters, we call it as microadenoma. It's more than 10 millimeters or 1 centimeter, we call it as the macroadenoma. But there is another macroadenoma, that's what we call as giant adenoma, in where we do see an overall volume of 30 to 40, more than 10 cubic centimeters, or 30 or 40 millimeters in width. While talking about the invasiveness, and what we see is what the pituitary adenoma makes is, it is the capacity to invade the surrounding structures. Basically, data comes from the neuroradiological or histopathologic investigations. As long as a pituitary neuropathology receives the specific small samples of the dura. If we don't receive, we cannot say that it is an invasive tumor. But basically, the data comes from the neuroradiology that I believe the neuroradiology and neuropathology are the two partners which you cannot divide them separately. For example, in every neuropathology report that I sign out, you can see a note coming from the neuroradiology. I personally look at the cases and look at the notes, which is written by the neuroradiologist. And if if there is a trouble, to solve that trouble, I directly call my colleague and ask him what he means by saying this is the finding of this one. So in order, they are inseparable for each other in terms of the diagnosis, whether this might be a brain tumor other than the pituitary, or whether this might be a pituitary tumor or a metastasis or elsewhere. Now let's go back to the invasive potential of the pituitary adenomas. The invasion of sinusoid and sphenoid sinus and the cavernous sinus is happening with a ratio of 25 to 55% of all pituitary tumors. So it is not very unusual. The other thing is, just remember the schematic representation that's next to the cellular diagram, and the supracellular extension of the tumor is not a criterion of invasiveness. While classifying the neuroradiology, the pituitary adenoma or the pituitary masses, we use either the Hardy's or the NOS classification. That totally it tells us the situation of the mass that lies either within the pituitary or around the pituitary or just a little bit far away from the pituitary fossa. So I will be mentioning about the details of the both the Hardy's and the NOS classifications. So how they define it, because that is quite important for the neurosurgeon to show to where to follow, which route he or she should be following while getting access to the pituitary area. Macroadenomas are in generally can invasive, but they are not often clinically aggressive. That means bigger the volume of the adenoma doesn't mean that more aggressively your adenoma is. So there is some confusing facts about the size and shape of the pituitary adenoma. What about the aggressiveness? What we mean by saying the aggressiveness is, in general, the aggressiveness, the data comes upon the clinical behavior of an adenoma. And we don't still know the exact number of the percentage of the adenoma because the series coming from the different the articles tells different ratios and different numbers. Exhibition of some atypical findings, such as the high mitotic activity, which has been proven by the Ki67, if it is a more than 3%, or the higher the ratio of the P53 immunopositivity, just gives us an idea about that aggressiveness of a tumor. Now, I will just give it a little bit on the next coming slide, how capricious the P53 immunostaining is, because once you apply the P53 immunopositivity, some laboratories says that our cutoff value is that if you just take the same sample, just give somebody else. And in a laboratory, they can say that this is not a well-stayed, properly stayed, this is not a properly the applied technique. In that case, since the inter-laboratory observation is not very set appropriately, this is a problematic issue still for the time that we live the same problems, not only in the pituitary, but in the different areas of the central nervous system. In generally, the pituitary, the aggressiveness is based upon the invasiveness of the tumors, macrogiant adenomas. For example, if you just look at the paracellular extension or the bone invasion, they act a little bit more aggressive. But on the other hand, although we don't see any high proliferative index, which is counted as the number of the mitosis in 10 high power field, or the number of the Ki67 positively staining cells, you might not be seeing all these with a relatively low the percentage of the P53. There are some tumors. When you are able to detect some specific features, characteristic features, they act aggressively. We know that. For example, this group is just being formed mainly the ACTH adenoma. Microadenomas act very aggressively. We know that. And we have a list which has been consisted of 6 different the forms of the forms or the histotypes of the pituitary adenomas. They act aggressively. Now the issue is what makes them to act aggressively. We believe strongly that this data will be coming from the biomarkers of the pituitary glands. Non-metastatic, that doesn't mean that pituitary adenomas metastasize, because if a pituitary tumor, an adenoma, has the propensity, affinity to metastasize either within the cranial wall or by using the the leptomeningeal seeding, in that case, it is no longer called as a pituitary adenoma, but it is a pituitary carcinoma. By just looking the ugliness of the cells under the microscope doesn't guarantee that you're looking at the pituitary carcinoma. What we basically need is the ability of that tumor to metastasize either within the central nervous system by the way of direct invasion or the leptomeningeal seeding, or by using the blood-borne metastasis to reach the places outside the CNS. Okay, now this is the invasion of the surrounding structures by pituitary adenomas complicates this neurosurgery. So this is a clue for the neurosurgeon, for the clinician, that this tumor that has the propensity to invade the surrounding tissue might be troublesome in the long term, the follow-up of the patient, because it has a higher likelihood of recurring. And now let's look at this, the difference, the meaning of the atypicality, invasiveness, and the aggressivity of the pituitary adenomas. Now, the pituitary adenomas can be classified as the data coming from the pathologic features, typical or atypical, or the data comes from the radiological findings, invasive or non-invasive, or the data might just coming from the clinical behavior, the aggressiveness or their non-aggressive adenoma. So in order to describe the difference between the aggressive, is it equal with the invasive or the atypicality? These are the questions. There's the three different terms. One means the aggressivity, the other data comes from the invasiveness. The third one comes at histologic features, atypicality or atypical or the regular adenoma. Now, in order to dig the secrets just behind the pituitary adenoma, there are so many working has been done, so many research has been done for the last, say, 60 years at best, but still we need the clear definitions that I just tried to show you at the top. We need still clear definitions, which is which. So while looking at the 2004 classification, it is in a small entity, in a small phrase, it says that there are two forms of the pituitary adenomas. One is known as the typical adenoma, the other one is the atypical adenoma. That these are completely different from the pituitary carcinoma, as you can easily recognize just reading that slide. Now, while looking at all these, a typical adenoma is been described as a monotonous histological features and with infrequent or the nil mitotic figures, and the Ki-67 proliferative index should be less than 3. In the case of the atypical adenoma, just look at the read, these the ones in these are bold and red. There should be a Ki-67 index which is more than 3 at the nuclear staining for the P53. In that case, you can call this lesion as an atypical adenoma. The carcinoma is a different story, because in that case, to call a tumor as a pituitary carcinoma, there should be either a cranial-spinal dissemination and or a systemic metastases, although not all of them may show cytological features of malignancy. Again, when I used to be a resident in the pathology department, whenever we just see a little bit of ugliness while looking under the microscope to the pituitary adenoma, if there's some, atypicality such as size and shape differences, and here or there, we easily call it as a pituitary carcinoma. I know this is wrong. This is not correct. In order to call a lesion as a pituitary carcinoma, we need a craniocerebral dissemination or a systemic metastases. Now, what about the invasive adenomas? Invasive adenomas are the adenomas which have a propensity to invade the cavernous sinuses. Let's remember the cavernous sinuses just lie on both sides of the pituitary fossa, and the bone either is phenated to the roof of the sphenoid sinus or the other body structures, or that's so. So in that case, if there is an invasion which is proven, which has been demonstrated clearly by the neuroradiology, we can call it as the invasive adenoma. Supracellular extension is not a criterion, because when you just during the time of the surgery, if the appropriate sample has been taken from the diaphragma, in that case, it is possible to find out the presence of some minor foci of infiltrative areas which are not accompanied by the recurrence rate of these tumors. So we just put it away in one place. So the invasion should be spotted either by using the MRI or during the time of the surgery, or with the histological demonstration of the tumor spread to the dura, bone, or the nasal mucosa. And you might be asking, the nasal mucosa should be remote from the pituitary fossa, But it is not surprising to see in the case of an invasive adenoma to gain access straightforward into the nasal mucosa and make its the invasion in that particular area. And sometimes during the time of the operations, we receive some, I would say, the samples. That's what we call intraoperative diagnosis or the frozen section. The neurosurgeon asks me, the neuropathologist, whether do you see any, say, samples taken from the nasal mucosa, any signs of the invasion of pituitary adenoma? Sometimes we say, yeah, here is the pituitary adenoma, although it's far away from the pituitary tumor itself. But in the case of an invasive adenoma, we can see these as a part of the process. MRI is the most common approach to classify the invasion. Nowadays, there are different techniques used by the neuroradiologists. Believe me, this is something that I've just said some days ago while discussing on, again, a meeting, that next time when I just have the opportunity to be born, I will be qualified as a molecular neuroradiologist, not a pathologist. When a neuroradiologist gives you the idea, you are looking to a jungle. As a pathologist, I'm just looking to a tree. And there's always a risk of barking at the wrong tree. But if you imagine that it's a part of the jungle that is being enwrapped by the radiology, and with the use of the molecules while defining and looking at the whole picture, the whole jungle, that will enable you to get some idea about the biologic truths, the biologic reality. We are at the time, at the platform that I lie in is too far away just reading the reality. NOSP and the Hardy grading systems are widely used. More or less, they just mean the same thing. This scheme is taken from the Antonio de Leivas, the article which was published in the Natural the Reviews of the Endocrinology in 2014. At the top level, you can see here is the Hardy's classification. It is very easy to understand. And so it's based upon the location of the tumor, of the pituitary tumor. If it is within the cella, you can see that the red spot signifies where your tumor is, the adenoma is. It is just beneath, within the cella. And it is very close to the cellar, the wall. In that case, we call it as grade 1. If it is expanding to all parts of the pituitary cella, grade 2. It is beyond the sphenoid side, in the sphenoid sinus, is called as grade 3. And it is bilaterally invading the surrounding structures that involves the cranial nerves and the carotid. In that case, it's called as a grade 4 lesion. Now look at the NOSPs, the classification. This is also more or less the same thing. The tumor lies within the cella, the tumor expanding on the body structures above the dura and expanding into the carotids, the arteries. And also you can see that not only the bony involvement or dura, plus the carotid arteries has been just enwrapped by the tumor. And that tells us the grades 0 to 4 in Arabic letters. This one is in Latin, but in Latin characters. But basically they mean the same thing. Whether you are just dealing with an invasive tumor and how invasive the tumor is, how invasive the tumor on its adjacent structures. So the invasive adenomas, the dural invasion is detected by the microscopic examination is not infrequent, which means we do see, as long as the pathologist receives the sample tissues from the dura. So if you just look at carefully, it is possible to find out the invasive spots. Therefore, this is not regarded as a constant indicator of an aggressive tumor behavior. So another interesting point is that I should be telling, which I did not place that on that during my the presentation, is for about a decade ago, when I used to be working in the Institute for Neurological Sciences, Marmara University. My fellow and my friend neurosurgeon just took the samples in the from the post-mortem studies which were done in the forensic medicine departments, and they just make the serial sections, and they found that there are some natural holes within the dura, just next to the diaphragma cella. That means what we think as the wall, the solid wall that lies this the fossa is not solid as we think. There's small holes. That means if we are talking about a tumor, the tumor can see that small pathways, the small highways, or the small, say, the motorways to go to use and straightforward just invade the surrounding structures. It doesn't have to dig the dura itself. By using the natural spaces, it can go beyond. And this was published for about a decade ago, and to the surprise of the neurosurgeons world. So that was a clause that I should have mentioned. I should be mentioning about that. The overall of the cavernous sinus invasion is for about 1/3 of all cases. Cavernous sinuses just lies on both sides of the pituitary fossa. And always we expect to find out the cavernous sinus invasion more in the macroadenomas than the microadenomas. That is an understandable issue. And in the dural invasion, if you just look at them microscopically, almost two-thirds, even more than the three-fourths of the cases shows a dural invasion, microscopic invasion. And the large adenomas also show more the invasive potential. That is, again, an understandable issue. And these were discussed in the biomarkers of the aggressive pituitary adenomas by Özgür, Mete, Shirin, Azat, and Silvia Asa. These are the guys that live in the Toronto, in the University of the Toronto. They are the students of Dr. Kovac. And aggressive pituitary adenomas with the diagnosis and the emerging treatments, which was again discussed by Antonio, Fabio, Luis Ciro from Colombia, Cusimano, and the Kalman Kovac. So this is to say that they have been just working on that field more than 40 years on a specific organ. Now, what about the a little bit more wording about the aggressive adenomas? Aggressiveness is described as the ability of a pituitary adenoma with their multiple recurrences and with their the resistance to the conventional treatment. In that case, whatever the histotype, whatever the histologic type of the pituitary tumor, we just call these lesions or the tumors as the aggressive adenoma. But this might be an ACTH adenoma, in other words, a corticotropinoma, or a somatotropinoma, in other words, a GH secreting adenoma, or a gonadotropinoma, FSH, LH adenoma, or say the TSH adenoma. If we are dealing with a tumor that shows the multiple recurrences and that is resistant to the conventional treatment, we call it as the aggressive adenoma. The question, what makes these tumors so aggressive? Why they are resistant to the conventional treatment? Why they show the multiple recurrence? The answer comes from the neurosurgery department. If they're unable to take it out properly, that's what we call it as a gross subtotal the resection. In that case, you just leave the tumor behind and it recurs. But you're quite sure that by just after the time of the surgery, looking on the neuroradiological the scans, there's no residual tumor, but it recurs for about 1 year or 2 years later. So there is some other point that we cannot see during the time of the operation and during the time of the follow-up course and follow-up trait of this group. So why they are resistant for the conventional treatment? What we understand by saying the conventional treatment is radiotherapy. We don't know. We don't know. They are either the larger makes a sense, or they are faster in growth, or they're either larger and faster in growth. This is what we call as the aggressive tumors, aggressive pituitary tumors. According to their clinical behavior, this is aggressive versus non-aggressive, is the description. Now, why and how do we diagnose a case as an aggressive pituitary adenoma? They show high rate of recurrence. They demand repeated surgeries. And the additional finding is they are resistant to the conventional treatments, which what I basically mean is the use of the radiation. The correlation between the classification is not proportional, means the data about the aggressiveness coming from the different series doesn't fit to each other in 100%. Pathologic findings do not always correlate with the radiological findings and clinical behavior. we say that this is a benign, this is a very silent, this is a very innocent looking tumor. But the neuroradiology says that it is invasive because it's some hemorrhagic foci, some necrotic foci. Clinically, they said this is aggressive. So there's a discrepancy between the histopathology classification and the clinical findings. And these are the confusing facts. Some typical adenomas may have an aggressive clinical behavior. For example, if their Chi67 mitotic index is more than, although it's less than 3%, they act aggressively. They do show high rate of recurrence. They are resistant to medical and the surgical treatments. So the proliferative potential is not the determinant of its aggressiveness. So, however, on the other hand, while talking about the atypical adenomas, they don't always follow an aggressive clinical behavior, or they can also be invasive or non-invasive. So you might be asking the question, why we call them as atypical adenomas, if as long as they no longer show an aggressive behavior. So the atypicality is not the equivalent of aggressiveness. They are different. Now, not all the invasive adenomas display pathological features to be considered as the atypical adenomas. So this is another finding. Although our tumor, the pituitary adenoma, is invasive, under the microscope, everything seems to be quite salient. So some of the non-invasive tumors behave aggressively. Again, it's another confusing fact. So the classification is based upon not only the pathological features, because we need the radiological and surgical findings to make a distinction between the invasive and the non-invasive potential of an adenoma. And aggressive and non-aggressive pituitary adenomas are just being based upon their clinical behavior. So the data just coming from different aspects: pathology, radiology, and clinics. All together, we just try to describe. So if we just go back to the 2007, this is a very famous lady. Originally, she is from Lyon, from France, with her the group. They've proposed a new classification saying that there should be different aspects while evaluating a tumor. Propose 3 subgroups based upon the radiology and the surgical characteristics, which designate the invasiveness of the aggressiveness of the tumors. That is to say, the difference between the non-invasive versus invasive, aggressive versus the invasive adenoma. So the aggressive invasive adenomas show a Ki67 labeling index more than 1%, and the mitotic activity more than 2 in 10 high power field, and with a description of the description of the P23 expression. So while diagnosing the aggressive adenomas, their distinction and definition are controversial. For example, if you just, they're different in the different the laboratories. When you just look at the Dr. Heaney's, the classic, the manuscript, it says they should be showing more than 10% of Ki67 labeling index. Pascal says that it should be 20-30%. So the diagnosis is always too late and too little, say Pascal in another the manuscript that's written in the same name, the same year. The Trula, Dr. Jacqueline Trula, like she's a retired lady, a pituitary lady, says that he just described the classification based upon the 4 major subgroups. 1B is described as a non-invasive but proliferative, 2A invasive, 2B invasive and proliferative, 3 is metastatic. Metastatic is no longer assumed as a pituitary adenoma, it's carcinoma. Now, the aggressive adenoma, how we should be calling these? Is it an unrecognized carcinoma? That makes the explanation. Or is it this tumor a premetastatic carcinoma of the within the cellular phase? Or is it an in situ carcinoma? Or an aggressive adenoma? Could it be a localized carcinoma? Or the tumor suspected of malignancy? We don't know the answers for that for the time being. But we know that there's some specific histotypes of the pituitary adenomas acting in an aggressive manner. For example, if we deal with a tumor that what we call as a Crook cell tumor, that's a specific form of the ACTH adenoma, in other words, the corticotropinoma, that you might recall that the Crook's hyaline degeneration is a kind of a microscopic description, which was forwarded by the Dr. Crook, is the presence of some eosinophilic material within each cell. ACTH cell is described as Crook, hyaline degeneration. But the tumors that has been mainly consisting of that kind of cell type is called as a Crook cell adenoma. If we are looking at a Crook cell adenoma under the microscope, we know that this will act very aggressively. If we are talking about a gonadotrope adenoma, it will act differently. If we are talking about a sparsely granulated GH adenoma or somatotropinoma, by definition, it will act very aggressively. Or a densely granulated lactotrope adenomas act very aggressively. Acidophil stem cell adenoma will act. Thyrotrope adenomas, some of the pleurohormonal adenomas, and silent adenomas of different types, as well as the null cell adenomas, by definition, we know they act very aggressively. So a large scale of pathological, ultrastructural, structural, and molecular studies tries to solve that problem. What is the idea behind aggressiveness and the tumor type is still unresolved issue after so many wording. Now, in 2017, a new publication by the WHO just come into action, and it was released for about 4 years ago. And if you just look at that group of the specific portion called as the pituitary neoplasms, discusses widely the impact of the potential biomarkers. So, for example, the apoptotic markers has been searched extensively. That is to say, the impact of the P23, P27, or P21, or the inhibitor cell cycle proteins like the COX-2, or the angiogenesis markers such as the VEGF, or the mean vessel density, numbers of the proliferating capillaries within the tumor, or CD31. That is a straightforward, the surrogate of the proliferating capillaries, or CD34, likewise the CD31, or the hypoxia-inducible factor 1 alpha. And they've all been searched. Matrix metalloproteinases, in other words, the MMPs from 1 to 12, proliferative markers, Ki67 or topoisomerases, or a galactin-3, or the pituitary tumor transforming factor, or the microarrays, or the pituitary stem cells, the microenvironment of the pituitary, or inflammation and the tuber linkage. And these are all being checked out. And the tumor heterogeneity, the famous entity known as the tumor heterogeneity. We assume that, we accept that the tumors, the pituitary tumors are the, the homogeneous tumors showing the same kind of the genetic material. Is that true? The recent study says that no, they are not the same thing. They are different animals. So this was discussed in a paper that I just wrote in the 2012. Just looking at that time slot, I just looked at 163 different the manuscripts, which just discuss the different aspects of the biomarkers. Well, it is surprising to see that there's not a clear cut, the outcome on that all research, other than the Ki67 and the other than the P53. And at that time, that was in 2014, the Antonio just wrote a letter. Altogether, we just wrote a letter about the microRNAs that was in fashion at that day, whether they have a straightforward impact in predicting the outcome of the tumors. The answer is, again, it is a promising field without the very clear-cut the findings. So the mRNA, the microRNAs are still a topic which is under very severe investigation. There are still too many things to be done in that field. Now, again, the very classical the data coming from the Dr. Assas, Dr. Assas and the Shireen's, the pathogenesis of the pituitary tumors. About the pituitary tumors, now we just came to a conclusion that transcription factors have a straightforward effect in the pituitary oncogenesis. This is to say that they originally, as I've told you, the anterior pituitary just is an extension of the expansion of the stomatium, in where there are the pertinent features of the oral ectoderm that shows a group of the transcription factors, which shows some differentiation in the way. To the right, you can see the Radke's pouch stem cells. If you're talking about the TPIT or the NeuroD and the beta 2, and they show a differentiation in the way of the corticotropes. Again, the same says stem cells under the transcription factors of the PIT1, they show a differentiation towards the somatotrope, shows a somatotrope differentiation. With the additional the transcription PIT1 beta, they turn into the lactotropes. This likewise coming from the origin of what we call as Radke's pouch stem cell. By the effect of these the various parameters, they differentiate into the gonadotropes. At the level of the somatotrope stem cells, they differentiate into thyrotropes under the control of the GATA2, TEF, or the GH receptor, or the PIT1 beta. So that is to say that while looking at these pituitary adenomas, we have to find out the effect of the transcription factors right now. Now, this is what the 2017, the Blue Book tells us about the impact of the transcription factors. This is to show you the recent data coming from the loose zeros. I myself, we just wrote this one and published in the 2015. Here on the left-hand side, you can see the different forms of the different histotypes of the pituitary adenomas, whether they are able to secrete the GH adenomas or mammotosomal somatotrope, or a mixed tumor that has the GH and PRL within the same tumor, or the adenomas which are able to elaborate and secrete the PRL, the lactotrope adenomas with densely and without the granulated, or with or without granulations, or acidophil stem cells, all has the same common transcription of the PIT1 on the immunohistochemical staining properties. If you are looking at an adenoma which has the propensity to be stained with the TSH, TSH is the end product, thyroid stimulating hormone. In that case, they are just being controlled by the PIT1 and the GATA2. If you are looking at a tumor that has the propensity to be stained toward the immunohistochemically by ACTH, whether densely granulated or sparsely granulated or Crook cell, they are under the control of that specific transcription factor known as a TPIT. And likewise, we have lists in that one. That is the key nowadays to understand the behavior of the tumors. Now, again, I'm not going to details. This is a little bit, what about the use of the oncogenes, tumor suppressor genes, and transcription factors? There is a correlation in between the ability to detect the transcription factors and the T tumor suppressor genes, such as the P23, has more or less has an impact in understanding, in determining the behavior of these tumors. So what we basically need is the further investigations of new oncogenes, tumor suppressor genes and proteins, and the transcription factors, which was described in the 2017 classification. Now, a little bit of information about Ki67 and P53. This is a trustable parameter. We are just counting down the number of the mitosis in that case, because while looking at the mitosis, a pathologist only is aware of the M phase, the prophase, telophase, oncokinases, and the metaphase. But we're no longer able to detect the G1S and the G2. But the Ki does that work for us. It just indicates that the cell is in the process of the dividing. P53, as I've told you, is a cellular antigen that involved in the cell cycle regulation controlled by the TP53, which is located on the 17th chromosome. And the higher the expressivity, the mutancy, and we expect a bad prognosis. But the problematic issue is a technical issue. Whatever you just get a positive reaction in one laboratory is not being accepted in another laboratory. So the recipes about the P53 staining is still problematic. The same is true not only for the pituitary tumors, but also in the other sites of the human body tumors. What about the use of the molecular markers? They're not fully justified. They are still under the way of we are just making a lot of research to understand these. So up to date, the histopathologic biomarkers in predictive the invasive adenomas yield no unconvincing results. So the biological markers for the aggressive manner of the pituitary adenomas is still being under investigation, but none of them just been accepted as being responsible for invasiveness. That is to say that we have so many question marks to be answered by the different techniques. So the poor problem about the pituitary tumorigenesis is we still have a lot of question marks in understanding the pituitary tumorigenesis. This is the involvement of the tumors in the pituitary, its ability to progress, and its ability to invade. The same is true for all the tumors in the human body, the human visceral and the non-visceral organs. Now, this is to say about this is all the things I'm going to tell you about the pituitary the tumors. Now, what about the pituitary carcinoma? That is a very small ratio. One of the two best the articles written about the pituitary carcinomas, in my view, is the pituitary adenomas and carcinomas written by the Dr. Scheitauer, the famous, the neuropathologists, and also the pituitary carcinoma written by the Beatrice Lopez, Dr. Scheitauer, and the Dr. Schiff. They described in details the qualifications and the characteristic findings of the pituitary carcinoma. In order to call a tumor as pituitary carcinoma, there should be a cerebrospinal and or systemic metastasis. This is the first thing. And they're almost in 2014, 150 tumors published to date in English-speaking, in Anglo-Saxon literature. They only form 0.1 to 0.2% of all pituitary tumors. So they show an extensive invasion of the brain by the pituitary tumor. We still do not know this should be regarded as a criterion. The criterion is their ability to metastasize by using the cerebrospinal fluid, or the extracranial metastasis by using the hematogenous, the problematic issue. And when we just look at the pituitary carcinoma with the metastatic foci, with the metastatic capacity, there mostly are ACTH producing tumors, or PRL or prolactinomas, or the TSH secreting tumors, some of which could be the somatotropinomas, or there's one case in the literature that the gonadotropin releasing factor, which is very rare, written by Dr. Kuchion, who is an original a Scottish fellow who lives in Houston. We just know him personally and sit down, have drank for about 5 years ago. The pituitary carcinomas arise from either the invasive relapsing adenomas or inactive adenomas. So there is no guarantee that whatever pituitary tumor you are looking at will act in a pituitary carcinoma way. You cannot foretell all these just looking upon their morphologic features. This is again taken from the Antonios, the classic, the manuscript, which tells how a pituitary carcinoma is the carcinogenesis is being followed. This is the normal pituitary. Here is the pituitary adenoma. That due to the unknown reasons, upon the effect of this and the control of different systems, we might be seeing an aggressive adenoma. This solid line tells you, again, without those transcription factors, some invasive, the molecular environment changing, and this makes this tumor as turning a pituitary carcinoma by metastasizing. Or the alternative pathway could be, this is a pituitary tumor, and you can see from the initiation point as an aggressive pituitary adenoma, which has the capacity to metastasize. We don't know which is true, but these are the theoretical aspects of how can a tumor show a dedifferentiation and make metastases in order to call the tumor as a carcinoma. Morphologically and structurally, or the microscopic evaluation, the pituitary carcinomas are not very different from the atypical adenomas and even the typical adenomas. Pituitary tumors, the carcinomas have been just looked at their the KI, the proliferative index. P53 oncoproteins, and they do show a low expression of the P27 in the primary tumor and its metastases. So they are different animals. Again, this has been discussed in, again, the Antonio's, the paper that the solid line tells us here is the normal autopsy pituitary gland. You can see the very low number of the pituitary leveling index of the Ki-67, an index. In the non-invasive adenomas, you can see from 0 to 3, the Ki-67 index in an overall area of the in an overall area of the 10 fields, the high power fields. In the invasive adenomas, you can see they just show a very the wide spectrum of changes. Although we call it an invasive adenoma, in some cases you might see in the 15% of the Ki-67, the propensity or the reflection, in where you might be seeing no Ki-67 positive cells. So can you rely on this parameter? The answer is possible, no. And since the pituitary carcinomas, you might be seeing no proliferating cells, the Ki-67 cells. On the others, you might be seeing a very high level of the proliferative index. The proliferative index sometimes, although you will try to rely on this, cannot guarantee, they cannot warrant that it's possibly makes up to make the differentiation between the different types of the adenomas in non-invasive, invasive, and pituitary carcinomas. So this is a problematic issue. Again, the pituitary carcinomas, their prognosis is very poor, and about 80% of the patients die within a period of 88 years. And very rare the patients just live with a long-term survival. We still do not know whether it's the success of the treatment modalities or whether the inborn and the ability of the cells to act in that way. So when you just look at these, make a comparison between the carcinoma and adenoma, definitely the carcinoma has a worse prognosis and they are quite radio resistant. And the two latest reports coming from the late 2014 tells us there are still problems in the related diagnosis, and there are still problems in biologic behavior of the pituitary tumors. So it is not easy to make a prediction about them. So the progress in the diagnosis and the classification of the pituitary tumors is still in the way of progressing. And in order to improve the differential diagnosis of the pituitary adenomas, there should a lot of things to be done with the additional data coming from the molecular environment. So how to conclude this presentation, which is so far almost 45 minutes, is the pituitary adenomas can be classified according to their pathologic, radiologic, and the clinical behavior. The pathologist says that whether we are looking at a typical or atypical adenoma. The radiologist says whether you're dealing with an invasive or non-invasive adenoma. The clinician tells that whether we are just dealing with an aggressive or a non-aggressive tumor adenoma. The WHO classification in 2004 says that we have two different forms of the adenomas. One is the atypical, the other one is a typical. 2017 says that there is only one type of the adenoma with different molecular environments. So the impact of the molecules have an effect on the behavior. While diagnosing the patients, while diagnosing each individual tissue, with of course the tissues of the patients, try to write down their immunohistochemical features, try to write down the transcription factors, and try to write down the amount of the proliferative indices and the impact of the P53 at the bottom line. Don't straightforward try to send out your report, sign out your report as the atypical adenoma, just relying upon the P53 and the Ki-67. Just write them as a note and try to keep an eye on your patients. So the atypical adenoma, which is defined as if the tumor is just having a more than 3% of the Ki-67 labeling index and or an excessive P53 immunoreactivity, doesn't guarantee that you are looking at an atypical adenoma. It should be noted as the baseline. So an invasive adenoma shows a pathological and radiological signs of invasion in the cavernous and or the sphenoid sinuses and bone and nasal mucosa. And the use of biomarkers in differentiating the aggressive adenomas has a limited place for foreseeing the prognosis. And as the another thing is, some specific histotypes shows the aggressiveness. Pituitary carcinomas are, although they are rare, they exhibit a cerebrospinal systemic metastases. They show a higher index of Ki67 and P53, and the protein expressivity, and they are resistant to the radiotherapy. So far, this is the whole thing about the that we just wrote it, and it was, that is, as you have seen, we just wrote some manuscripts about their behavior, the pituitary adenomas, and this is a group that I have been working for the last 10 years, that we produce at least the 8 different manuscripts. And so in order to have an idea about the clinical the behavior, about the invasive and the aggressiveness of the pituitary tumors, I think we need to make some more research based upon the data coming from their molecular environments. This is not true for the pituitary adenoma. This is true for almost all parts of the pathologic neoplastic proliferation. This is what we say as for example, when you step into the Dr. Kovacs office, he has a very small office. Their building is just coming from the at least 80 years old. They're in the Michael's Hospital. The first thing you see on the board is, on the wall, is a small piece of paper, A4 type of the white paper, and there's a specific saying which says that science is an endless and the frontier, never-ending frontier, which means, although he is, when I just met Dr. Kovac, he was 91 years old, but he was trying to find out what is the reality behind that. At the age of 90, just keeping that level of curiosity, that level of intellectuality, makes these type of guys, as the extraordinary guys. The same is true, my dear friend, is valid for Dr. Scheitauer, that we lost in the 2010. It's almost 11 years. And there were the two friends, close friends, after meeting each other in 1988. And I asked Dr. Scheitauer where I can learn the pituitary. He told me, I will send you straight forward to the Dr. Kovac's office. This is the story how we met each other. By the way of that, we are just met. I met with the Antonio, the Dr. Ciro, and Dr. Cusimano, and years, and Dr. And Fabio. Actually, Fabio is not an MD. He does everything in the office. So this is the whole thing I'm just going to tell you about the invasiveness, about the aggressiveness, about the carcinomas of the pituitary gland. I did not mention, and I didn't mention anything about the normal classification of the pituitary adenomas, but their clinical behavior, their aggressivity, and their invasiveness. Okay, thank you for your attention. If you have any questions, I'm open to answer all the questions you will be asking.
Thank you so much, Professor. We really appreciate you taking your time and arranging this lecture for us. It was, as always, very dynamic and informative presentation. We know there are even much more details about this topic, but you explained it and summarized it very well and very clearly, I believe. And we can take questions from all the participants, I guess. Feel free to write them into the chat box.
I think we have a question in the chat. I can read it if you want. Jason said, great lecture and very informative. I would like to ask the professor, according to your opinion, where should research focus on the shine some lights on the topics that are still undecided for pituitary tumors?
Yeah, that's a good question. So let's go back to the slides that I've shown you about the pituitary markers, the biomarkers. And when I just wrote this article, that was the 2012, and I'm also just keeping an eye on how the different aspects is being the research. That these are the things. If you just look at all these potential markers, that I wrote a second one and a third one about the biomarkers. Since we don't know the exact details of the tumorigenesis or the oncogenesis that I mentioned last year in the second semester, we still do not know the exact mechanism of the cancer formation of the carcinogenesis, or better to say the oncogenesis. The larger issue is the tumorigenesis. All these aspects that you read on that transparency is quite true. There's a lot of things to be done in the apoptosis. You know, the meaning of the apoptosis is the programmed cell death, which has been controlled by the genes, by the specific genes. And that includes the P53. You know, you all recognize that the P53, which is controlled by the TP53, is a gene that controls if there is any escape during the time of the mitotically active the cell. If there is some or disorganization, it stops the cleavage, the mitosis, just let the endonucleases to come into action and repair. If there is not a foolproof repair, in that case, the apoptosis genes come into action and just gives rise to the apoptosis. This is another field, a most important, one of the most important topics. If we are able to manipulate the apoptosis, if we as the mankind are able, in terms of the medicine, to manipulate the apoptosis, you can guess how successful we'll be while dealing with the tumors. It is for us to keep an eye on the mitosis or mitotically active tumors to say, no, you stop here. I will just try to send the apoptosis or the genes controlling the apoptosis. This is a field. Or the inhibitor cell cycle proteins, which has been mainly just one of these. The stereotypic example is the COX-2. This is another area. Angiogenesis. Angiogenesis means the proliferative cells, proliferation of the endothelial cells and the capillaries. Now, in here, you can see the MVD. The abbreviation tells us the microvessel density. This is the number of the blood vessels. Now, it is surprising, though. When you look at a tumor that is lying next to the normal tissue, what do you expect is the number of the proliferating cells should be in excess than the normal tumor, the normal cells, the normal tissue. And by theory, the more the number of the cells, the more the number of the oxygen and nutrients they need. That's true, isn't it? But surprisingly, in the pituitary adenomas, the number of the capillaries are much more less than the surrounding normal tissue. We don't know why it is, but it is so. Now, this area needs another impact in the field, in the field of the research. Metalloproteinases. This metalloproteinases is a group which in the central field they have the zinc and they have the ability to form the backgrounds. And in that strict sense, less than amount of the metalloprotein, higher the amount of the metalloproteinases, the lower the conductance, the strictness of and tissue that makes the cells to freely proliferate and make some distinctive the behavior. This is another, if we are just be able to control the metalloproteinases. The proliferative markers, as the name it tells us, proliferative ability of the cells tells their ability to grow. So how can we control this? By using the chemotherapeutics, we are trying to kill the cells, to stop another PTTG, another interesting pituitary transforming tumor growth factor. This is another group, is another specific molecule that controls the proliferation of the tumor, the pituitary. That's why there's a P at the beginning. Microarray, it's an unbelievable large group that involves mainly the microRNAs. The pituitary stem cells of any kind of the tumor stem cells. We all know nowadays that in a proliferating the tumor, there always a stem cell, the cancer stem cell that harbors that that forms the major pluripotency, while just giving rise to the sister cells in a part of the tumor itself. Pituitary stem cells or in large parentheses, stem cells is an area in where you can apply the stem cells of the neural stem cells or the visceral stem cells or inducible stem cells at a large group. Microenvironments, if you just take the samples, just put into different, say, the in vitro conditions, they act differently. What makes them to act differently is an area of which has been the lasting for many years in the environmental, in the experimental pathology labs. Inflammation and tumor. Most of the data in relation with the tumor molecules come from the inflammation. This is something I learned during my time as a medical student, that I worked in the pathology laboratory. My boss at that time spent his time mostly on the amyloidosis, which is in where you can see the impact of the molecules coming from the inflammation. That includes the vascular, the endothelial factors, that comes from the hypoxia-inducible factors 1 alpha, and all the cytokines, all the lymphokines. They have a straightforward effect in inflammation as well as in tumorigenesis. Tumor heterogeneity is a genomic. The abnormality nowadays, we all know that, and the pangenomics, that includes the epigenomics plus the RNA omics plus the DNA omics, is under the very a straightforward investigation in the different laboratories. So there is not a straight only one answer for your question. It is a very complicated issue to find out which is true under which circumstances, which are being described, not only the tumor itself, the cell itself, but it has been controlled and defined by a large group of the external stimuli. That is what we call as the effect of the microenvironments in any kind of the tissue process or the pathologic process. Is that the correct answer to your question, Brock?
It wasn't my question, but a friend from Greece, I think.
Good.
So I have another question. Does these biomarkers can be a guide for the treatment?
Yes. Answer is clearly yes. And this is what's been done nowadays. This is what we call as the targeted therapy. If we are able to detect, if we are able to prove the effect of a biomarker, and if we are able to produce some specific, say the treatment modality or the treatment, say the chemotherapeutic agent, it will be our success. An example, for example, we all know that there is a proliferative, the function of the vasculature. More the number of the endothelial cells, more the number of the capillaries. That's correct, isn't it? And we have, based upon this information, we just produce a specific say, the chemotherapeutic known as the bevacizumab. And this directly kills the endothelial cells. This is what we call the targeted therapy. The answer to your question is yes. If we are able to produce a specific therapeutic, say, the agent that destroys that specific biomarker. If it is an inhibiting one, we have in that case to promote this one. If it is, say, an activating one, in that case, we have to suppress it specifically. And this is what we call the targeted therapy right now. And this is the answer to your question just comes again during my studentship days. During the summer period of the 5th and 6th year, I passed some time again in Dalhousie, in the University of Dalhousie, in the very eastern part, in the eastern shore of the Canada. That in a lab, my boss, the Turkish boss, was there, Alev Güçlü. He was just working on trying to find out the specific the antibodies which are just being directed against the rat renal cell carcinoma. By the way, that he just take the the small samples of the renal cell carcinoma, injecting it to the other tumors, to the other, strain, in order to get the specific antibodies directing against the renal cell carcinomas of the rats. Then he just tried to conjugate, the nitrogen mustard, in other words, a chemotherapeutic, so that he just produced a specific a drug named as the magic bullet, like güdümlü füze. When you just fire it up, it straightforward goes to the renal cell, and with the end, it has the tremonome, or in other words, nitrogen mustard, that in theory should be killing that specific the tumor cells. But in fact, the data coming, all the data coming from the experimentation show that when you just produce these specific antibodies, they also produce, without knowing, without intention, they produce some preventive or the blocking antibodies. When you inject that antibodies in order to kill the tumor cell, they rapidly proliferate, because the blocking antibodies just block the receptors of the tumor cells. So the specific antibodies that carries the nitrogen mustard just easily missed the targets. So there are too many questions that needs answers. But as long as you produce the questions in your mind by using the scientific methods, it's possible to find out the answer for each specific question. But it takes time. It needs your energy. It needs the targeting. It needs, what we call as the idealism, but scientific idealism, not the other one. Okay? Any other questions?
I think there's 2 more questions from
Where are they?
In the chat box. One of them is, does dural invasion affect the prognosis?
Just as how can I access to the questions? I don't see anything.
I think if you click on Zoom.
Yes.
Zoom window.
Maybe you can stop sharing, Professor.
Okay.
Return to the main.
Let's do stop sharing. Okay. And where the questions participant chat. Yes.
In the chat.
Chat. Now, which one is the first one? Okay, thank you from Mexico. Hello from Mexico. Thanks for invitation. Hello to great, informative. Hocam, dural invazyon prognozu etkiliyor mu? Answer is no. And that was asked by Sedat Üçer. Cerrahi sonrası radyoterapi tümörün atipik ve atipik patolojik önlenmesi karar veriliyor. Cerrahi sonrası radyoterapi tüpek no, that's not the answer. The answer, while just defining or determining the impact of the usage of the radiotherapy mainly comes from the ability of the invasive potential and the aggressiveness of a tumor. If your tumor, the pituitary adenoma, is an aggressive one, which means it is recurring, multiple recurrences, in that case, there should be a dichotomous area, whether you should be surgically removing the tumor for the second and third time, or whether you should be using the irradiation. That's a questionable issue. And it's totally dependent upon the data coming from the different series. But the typicality and atypicality is not the answer for the usage of radiation therapy. That is, the answer is no. And Sedat Uçar, is that okay? Did you get my answer? I don't see where this Sedat is. Sedat. And Burak asks, when giving the decision on
Yok hocam, onlar aynı.
Aynı? Okay, yeah, the one is written in English. Does dural invasion affect the prognosis? In that case, if we are talking about specifically the cellular diagram, the answer is no. Yes, Sedat says yes, he got the appropriate answer for his question. Any other visible questions? Dandy, Ethiopia is here too. Hi, Dandy. Walter Akrane is with you. Hi, Walter. Arabic, hi. Mexico, hi. Okay, these are all the questions.
Can I ask one question?
Of course, go ahead, straightforward.
In which case, the selection method, the operation, maybe you're told about it, but I connected late and I want to ask about that.
Yes, ask your question clearly so that I can understand.
When we can treatment like operation and when we start in radiation therapy, I ask about that.
Oh yeah, what is the indication for radiotherapy?
Yeah.
Correct. Yes. If you are dealing with a tumor, a pituitary adenoma, that shows multiple recurrences. In that case, we should sit down and think about either for a second surgery or a third surgery or an irradiation. This is a dichotomous issue. This is a problematic issue which should be given up by a committee or a consensus the meeting. Let me tell you one thing about the while dealing with the pituitary adenomas. Since the pituitary adenomas are insidious in their in their behavior, they are quite say an innocence. They are not very the aggressive tumors. Only small components, a small count, the small the percentage of them, the ability to show the aggressiveness. We should be looking at the first of all to the histologic features. We have to look at the Ki-67 and the percentage. That's what we are doing. The secondly is we are just looking at the P23, you know, the say expressivity, and we have to look at the radiology, whether there is a bone infiltration, whether it is the cavernous sinus infiltration. Then we should sit down and think about that. What will be the next step for us to? Is it time for the irradiation or not? Is there, which one will be more beneficial for the patient? Radiotherapy or the second, let's say, operation, or the third operation? Now, this should be done in terms of our colleagues to make a result, and to talk about which will be the more beneficial for the patient. For example, irradiation by itself has some adverse reactions. We all know that if ever just we irradiate the patients, now let's give me another the way of the example. If we irradiate the patients called as atypical meningiomas, which are known as grade 2, I'm talking about a different tumor. At the time process after the irradiation, we all do know that almost 15 to 31% of cases, there is an accompanying tumor originating other than the meningioma at the points of the irradiation. The radiation by itself has its adverse reaction. It doesn't just kill the tumor cells, particularly well-differentiated tumor cells. It also induces the oncogenesis of other tumors. Do you read my lips? Because irradiation has two ways of action. One is just breaking down the DNA. By that way, it kills the cells. The second one is the hydrolysis. So the water amount within the each cell, say in that case, specific cell, each cell has an amount of a volume of 15 femtoliter. And it involves a certain amount of the water minutes. When you just pass the high energy dependent energy, the rays, it hydrolyzes and gives out the hydroxy and these freely floating H's, which are just being entrapped by the superoxide mutase. So it is not always a good thing to irradiate. The irradiation should be one of the last steps in the way of the treating the patients. Now I'm just going back to the pituitary adenomas, the question you just asked me. And the irradiation, the time for irradiation, always we just keep as the last step. Resurgery and chemotherapeutics should be the other things, alternative pathways, if there is a strict need for an expanding tumor with the features of this, say, the invasiveness, with the features of this clinical aggressivity by the multiple recurrences. In that case, we can be thinking about of the irradiation. Now, lately, during the last, say, 20 years, the field of irradiation or the radiation oncology just started using a widespread way of taking the advantage of different the different particles, alpha, beta, theta, and the particles nowadays they're just using an enormous amount of wide spectrum of the radiation modalities. So in our the weekly conferences, in where we talk specifically on the Friday in the afternoons, in the afternoons specifically dedicated for the pituitary tumors, there's a small piece, a little place for the radiation. But if the radiation oncologist starts talking about the different way of the oncology, the radiation modalities, it is nowadays possible to radiate the patients just two steps before the last one. Yeah, everything is changing very continuously in the field of medicine, in the field of technology.
May I add something here about the radiation?
Of course,
Please.
go ahead.
It's not nice to irradiate close to the optic nerve. You may destroy it.
Yes, this is something else, and just depending on the location of the pituitary, just remember it is just located in a small fossa. It is 150 to 300 milligrams, 1/3 of a gram. We are just talking about a very small, a tiny, a minute, a subtle in the organ. Just above there is the optic nerve. A little bit above we have the paraventricular nucleus, supraoptic nucleus. On both sides, we have the the carotid sinuses, which are just being donated with the different say cranial nerves. When you just take into consideration, there might be some disadvantages point of the irradiation. But as I told you, nowadays the radiotherapy, the instruments are so extensively modified that just remember what the doctor, our neuro-oncologist tells us, just gives us, they are just talking about the stereotactic, the brachytherapies, that they're able to find out a small spot, and without any penumbra associated, they can destroy the target tissue. But on the other hand, once you destroy the optic nerve, it is a no return, and the point, and your patient might be free of this pituitary tumor, but suffering from the blindness. Yeah, you're right, Katya. Of course. Any other questions?
Thank you very much for your
Welcome. Was that the answer to your question? We can sit down and discuss about the point of the place of the radiation in the pituitary adenoma. That covers the whole session.
Thank you so much, Professor. I guess we have no more questions in the chat box.
Okay.
And on behalf of all, I also would like to thank Dr. Pantelli for supporting us from day one and joining us tonight. And I'd like to give the virtual floor to her in case she has anything to ask or add.
No, I enjoyed it really. I enjoyed it very much. Your lecture, Professor. There were things I did not know or remember, but I should not admit it in front of students probably. But my students know already that when I don't know something, I tell it. There were some things that you said in your lecture that slipped my attention. So thank you very
You're
much.
always welcome. You know, my door is always open to everybody. I don't keep the secrets for myself.
Yes,
Our lifespan is so short. Whatever I know, just will be. You know, there is a very well-defined quote which says, "Bilgi, sevgi paylaştıkça büyür." That is the main idea. You don't take anything with you into the coffin with you.
Yes.
Everything is changing.
I agree.
And I always tell to the people that, although you feel yourself as fully proven the professor, and one of the medical students in the first year asks you a question in front of a whole, this classroom. At that time, you are all aware that you know nothing. You know everything about nothing.
It has happened with some of the students I see here today.
Hopefully you enjoy it.
Where did this idea came from?
Hopefully you enjoy it. If you have any questions, you can write to me. You can just, my door is always open, but nowadays for the last 6 months, I'm just working at the home office. Everything has been done in my office, in that room, you know, all the lectures, you know, all the discussions, international or national, or the, you know, the questions, and or they say the docentic dossiers, everything is being done in that room. Okay. Thank you for everybody for attending the course.
Yeah.
If you have any questions, just write to me.
Yes, I would like to again give my thanks to our two inspirations here with us tonight, Professor Aydın for the wonderful and informative lecture. It's always lovely attending your sessions, and we're always looking forward to attending your other talks with your other hats, explaining other interests that you like and just indulge in. Another thank you would be to our Dr. Pantelli for being with us tonight here, supporting us always. I'd like to add a fun fact here. You are the one who made me go into neurosurgery and choose it as a specialty and just go on with it. So thank you again. Thank you all to everyone who saved us a place in their schedules and stayed till the end. We would love to and can't wait to see you all in our upcoming events as well.
Okay, thank you guys. Thank you. See you soon. Bye-bye. Take care.
Thank you so much, Doctor, for your presentation. And thank you, Water Turkey, for your invitation. It's a great fun and seeing.
Okay.
And well, we salute you from Mexico.
Okay. Say my best regards to Mexico. Okay. Bye-bye. Bye-bye, guys.
Goodbye.
Professor.
Thank you, everyone.
Can I say hello to the Dan degrees?
Yes, of course, Michelle.